Yes, so we are going to be talking today about some work that we just recently published last month actually, in molecular systems biology, and this is essentially a mapping of the whole viral proteome. And what we're going to have three people speaking. So this first will be Andrea talking about the modeling aspects of this work. Then I'll be talking about what we call the coverage map, which is how we view these models. And then Neblina will be talking about the system that we use called Aquaria to interact with the 3D models. And then I'll finish up with the demo of the modeling part itself. So with that, I'm going to hand over to Andrea to walk us through how we modeled the SARS-CoV-2 proteome.
Okay, so let's switch the screen sharing, right, Seán? So you need to, yeah.
Mm-hmm. Yep.
So just a moment. Okay. So what we wanted to do is to be able to show for every protein sequence that is contained in the proteome of SARS-CoV-2, to be able to show all related protein structures. So you can find any structural information that is available out there. And so we compared all these proteins against the whole proteome that has been structurally determined. The technology we've been using for that is the technology we have been developing over quite a time for the tool Aquaria, which we maintain. And that development began in the 2000s, where we already had the plan to show for any given protein sequence all related protein structures, and really relied on Maxom, which was one of the first profile-based alignment tools, and which was the tool behind the database HSSP, which shows for every protein structure all related protein sequences. And since we wanted to turn that around and show all related protein structures for all sequences, we also turned the name around. So that's why our database is called PSSH. By 2010, the databases had outgrown the capabilities of Maxom, and we looked for a tool that was faster and also more sensitive. And so we looked in the protein modeling tools and found hijblitz as one of the fastest and also most accurate tools for aligning sequences to protein structures. The reason why hijblitz is fast and accurate is because it is based on hidden Markov models. So for a given sequence, you're looking for first a hidden Markov model is generated, so a sequence profile, but it also uses a neat trick of converting this hidden Markov model into a kind of profile sequence, which makes a rapid searching really easy. And so this is the technology we're using in order to generate our protein sequence to structure alignments. How does it work? How does the workflow work? What we do is we use the database of HMMs that hijblitz and the hijsuit provide. We take all the PDB sequences, all unique PDB sequences that you can find, and scan these against the known proteins to build the profiles. And thus we get a HMM for every PDB sequence. And using hijsuit, that gets turned into a searchable database of HMMs. And then if you come in with a query sequence, or what we pre-calculate is for the query sequences, we also generate HMMs of those scanned against the uniclass proteins. Then you have a query HMM, which you can scan against the database of PDB HMMs. This generates the database PSSH2. And we also validated this tool to also find out which parameters are best. We did that by using a database of structures to structure alignments, which is based on a purely structure alignment mechanism. There is no sequence information in there. And we took all the proteins that have at least 30% structural overlap as true positives. And using that, we were able to balance precision of the match against recall, and decided that we're going to include all the proteins with an e-value of 10 to the minus 10 or better into our database. In 2020, since Helicase has been further developed, we redid our validation and checked that the 10 to the minus 20 cutoff still works reasonably. Since unfortunately the COPS database has been discontinued, we now switched to CATH, which is also a structure comparison database, and took as a true positive all the structures that have the same CATH code as a given structure you search with. And there we could show that if you take a e-value cutoff of 10 to the minus 10, in that bin around 10 to the minus 10, you still have 95% of the matches approximately that are really true positives. And if you take all the matches that are 10 to the minus 10 or better, then we have a cumulative precision of better than 98%. So if we show a structure as this is related to your sequence, you can be pretty sure that it really is related to your sequence. And coming back to SARS-CoV-2, we did this whole process also for the sequences of SARS-CoV-2 as soon as they were available. So they have been scanned against the 180,000 PDB entries, and we arrived at around 2060 sequence to structure alignments, which is great. But here I hand back to Seán, because now he explains where the problem lies and how we solved it. And I need to stop screen sharing.
All right. Okay, thanks, Andrea. Okay, my screen is coming across. Yeah, so as Andrea mentioned, we ran this process, and we now have over 2,000 structural models, which nearly doubles the amount of structural information that's available beyond what's experimentally determined in PDB. So on the one hand, this is fantastic. This gives us a wealth of new detail about the structural states occurring in SARS-CoV-2 proteins, but it also gives us a new problem, which is how are we going to make these 2,000 models accessible and usable for people? And my whole career as a structural computational biologist has been driven by the idea that structures are something really worth looking at. And we're all aware of this iconic image showing the value of looking at the structure that gave us this great insight into how information is stored and replicated. But of course, in the SARS-CoV-2 proteome, we've got proteins that are much more complicated than that tiny little piece of DNA that was shown there. And so the challenge is how to make these this really large amounts of structural information accessible and usable. And so for this, we developed what we call the coverage map. And so the concept is really driven by the genome organization. So many of you may know that the SARS-CoV-2 proteome is comprised of approximately 3 more or less equal portions: polyprotein 1A, 1B, and the final portion called the virion and accessory proteins. And we use these proteins essentially as a way to organize the models. And so to sort of guide you through the different aspects and different elements of the coverage map, I'm going to choose one protein. Particularly, I'm going to look at NSP3, which is actually the longest protein in the proteome. And this, as shown here, represents how we show matches that we find in one small region of NSP3. And as we see, in this case, we have nearly 400 structural models matching just to that portion. And so now to introduce you to the visual vocabulary that we're going to be using, whenever you see a structure, whenever you see a black mark in the system we call Aquaria, that indicates that that residue is in the structure is actually different to the sequence of the corresponding aligned position in the SARS-CoV-2 proteome or protein position. So that's the case here because what we have is a structure from a related virus. And as we see, it's only 23% identity. So the concept here is to give you a clear and immediate visual sense of the accuracy of the model you're looking at. And of course, most of the models are going to look like this. Most of the models have been derived are from related viruses, but increasingly over the last year, we've been very busy in structural bioinformatics and biology. We've determined now many structures, just over 1,000 when we did this study, that look like this, where you see immediately there are no black marks. That indicates this is a structure actually for exactly for the SARS-CoV-2 sequence. So when we have a structure like that, we use that, obviously the best matching structure as the example structure to illustrate the 400 or so models that are here. But keep in mind, again, most of them are actually going to be lower identity than that. Now, to make these models more interesting and to we also peeked into those models, in this case nearly 400, and we looked at them. And what we found in this particular region, which is called the macrodomain of NSP3, is something quite intriguing, which is that many of those structures actually are not viral proteins, but a group of them are actually human proteins. And what this is saying is that this portion of the NSP3, the macrodomain, actually mimics these human proteins. That's to say, it has a sequence similarity that is detectable by the alignment methods that Andrea just outlined. And these proteins all share a common set of functions related to a particular post-translational modification called ADP ribosylation. And in fact, what they do is outlined in the paper in more detail, but briefly, what this set of proteins do, or the most likely mimic function, is the removal of ADP ribosylation modifications that are made to proteins. And this is actually part of the virus's mechanism of avoiding innate immunity. So returning to the NSP3 sequence, and we see that there's no single structure that matches to all of the sequence. In fact, all we have are regions, and typically domains that are matched by various structures. And the second best studied domain is a region called PEEL-PRO. And this is a domain that is also has some interesting portions to the structure. In this case, when we look at that set of, in this case, 65 structures, we find no mimics, but we find that some of those 65 structures are found in complex with human proteins. And so this is regarded as evidence for hijacking of those human proteins. And in this case, all of the proteins that are found in complex with the PEEL-PRO domain are things like either ubiquitin or ubiquitin-like domains like ISG15. And so what this protein actually does, it's a protease, actually cleaves the N-terminal portion of polyprotein 1A, but after doing that, it then also cleaves ubiquitin and ubiquitin-like domains from host proteins. So again, this is part of unraveling the host's immune defenses, is by this action. So that's part of the reason why it's been widely studied. So this is a de-ubiquitination or de-ISGylation activity. That if we could block this activity again, we would be able to block or certainly inhibit the replication of the virus. Turning again to the NSP3 sequence, we see there's these regions where we don't find any structural matches, and these we call dark regions. We had a paper some years ago examining these regions in more detail. What's interesting about them is where we can say that the way the modeling is done, it's not that we don't know the structure of those regions. Actually, what we know is that these regions of the viral proteome are not detectably similar to anything that has ever been solved by experimental structure determination. So these are truly unknown in terms of structure. And these are ripe regions for more advanced modeling methods such as AlphaFold. Anyway, for our purposes, they are regions we just left as what we call dark regions on our coverage map. And so that's sort of how the map starts to look when we show when we extend this modeling to other proteins. And if we step back now and look at the whole proteome broken into these three regions I mentioned earlier, it looks something like this. Actually, one of the first remarkable things for us was the fact that this whole graph fits on a page and is readable, which was fortunate for us on one hand because we were able to print that as a single page figure of our publication. But it's unfortunate because what it says is that these are all of the hijacking and mimicry interactions that we have evidence for from structure basis. And there's actually not that many of them. We know indeed from many other kinds of experiments that there are more of these modifications and interactions that are known to occur, but they have not been captured in not only for SARS-CoV-2 structures, but for anything related to SARS-CoV-2 in terms of structural information. And there's also another kind of mimicry, another kind of event that I'll get to in a second. But first, I want to just focus in on one of the regions and the very first portion of the polyprotein 1A. This is NSP1. And so what NSP1 does is, I don't know if the audio is coming across there. NSP1 actually is the very first protein in the RNA genome. And immediately upon being translated, its action is to block the translation of host proteins and to start preferring the translation of viral proteomes. So from this point on, it starts to hijack the machinery. So this is a really clear example of hijacking, in this case, of the ribosomal machinery itself. So in addition to that, so this whole page is available as an online resource that you can go to, and in an online resource you can click on any of the states here, and it'll tell you more information about those states. You'll be able to access all the 3D structures for those states. So I'm just going to try to give you a bit more detail about the this macrodomain mimicry. This, I think, is a very interesting and perhaps slightly underexplored aspect of the viral proteome, that it mimics these ADP ribosylation-related proteins. The two proteins that are most strongly mimicked are both related to what's called the erasure of ADPR, so it's the removal of ADP ribosylation. This is more or less the opposite of what we think of as PARP, so the commonly known PARP-related proteins. They add ADP in multiple copies of it, so they add polymers of that, and these are called demyelation, actually. So they remove the very last of the ADP modifications, and so the interesting implication is that actually the PARP inhibitors effectively would act in the same direction as the virus. So the virus inhibits the addition of ADPR, and PARP inhibitors also inhibit the addition of ADPR. So PARP inhibitors may in fact exacerbate COVID-19, which is a new insight from the study that does not seem to have been noted, although I would say it's strongly supported from the literature. So there was another mimicry that we found, and this one is even less described in the literature, although it's actually supported by stronger evidence. So we have here an e-value of 10 to the minus 53, which is fairly strong evidence. So that and these proteins, these are occurring in a region of the viral proteome associated with viral replication complex. And this is NSP13, which has a helicase functionality. And the proteins that are potentially mimicked, these are human helicase proteins, but there's nearly 100 human helicase proteins, but only 3 of them have been recognized having significantly similar sequence identity to be potentially mimicked. So presumably it's specific functions associated with these helicases that may be being mimicked, and then functions associated with them being hijacked. The strongest one being UPF1 and the IgHMBP2. This one I think is particularly interesting as it's a protein associated with switching of the immunoglobulin class. And that's actually a function we've seen clinically is disrupted in COVID patients. So this may be a mechanistic explanation for that disruption. And I think this is a mimicry that really is worthy of further exploration. We have done some subsequent sequence analysis studying it up, but following it up, but I think a lot more could really be done here. So I'm just going to skip past these slides and just, okay, I'll make this point about this is now a summary slide we did showing the total hijacking, and the point here is we looked across the whole coverage map, and we looked at all of the cases where we have direct structural evidence of hijacking. This is the NSP1 that I mentioned before, and we're showing it bound to one of the a ribosomal structure. We note that it's a tiny little fragment of NSP1 that has been structurally determined as binding. Actually, I think in the structure, the rest of NSP1, which isn't a very large protein, but the rest of it was actually present, but it was not too disordered to be resolved in coordinates. But it's really just a tiny fragment that actually, where we have this direct structural evidence of interaction. And actually, if you look across all of the proteome, the structural evidence we have for interaction is really summarized in this figure in terms of the actual the residues that are directly in contact with human proteins. Overall, it's a tiny fraction of the proteomes, around about 6 to 7%. And again, we know that most likely a much larger fraction of the proteome is in direct contact at some stage with host proteins, but these are the only interactions that are captured. Some of these interactions, by the way, are really minuscule, as we see here. These are really tiny fragments of viral proteins. These are the parts shown with saturated coloring. These are really on the edge of what we would consider to be known structures. So that's just indicating that although we see quite a few of hijacking, these are shown here in the gray coloring, that actually it accounts for a very small fraction of the proteome. The mimicry is also a small fraction, again around 6 to 7%. And finally, I'd like to draw your attention to this third kind of interaction that we graphed in the coverage map, and these are shown with green coloring. And these indicate primarily interactions between viral proteins, so binding between two viral proteins, or binding to things like inhibitory peptides. So these are experiments that are done to test whether or not these could inhibit viral growth, or things like binding to antibodies. Again, so these are not indicating normal interactions with the host, but these are interactions that are being tested experimentally. And so we just use the green color to summarize all of those. And I'd like to just focus briefly on this upper right corner here, indicating what's going on. What this is telling us is that the NSP7 and NSP8, as you see, there's 35 and 34 of both of those proteins, structures in each case. And of those 35 and 34 structures, 32 of them, almost all of them, have both NSP7 and 8 in the same structure. So they have NSP7 and 8 bound together. There's only a few cases where NSP7 or 8 occurs by itself. And of the 32 structures containing both 7 and 8, 21 of them actually have an additional NSP12. And of those 21, 15 have viral RNA. So what we're doing here is we're seeing structures that form the viral replication and translation complex, which involves not only NSP7, 8, 12, but also 13 and also NSP9. And so we sort of see it assembling at various parts of this graph. So these are what we call teams. And there was basically one team here associated with replication and translation complex. And there was a second team associated here with NSP10, 14, and 16 that ultimately also should form part of the replication complex, but that complete interaction has not yet been captured in any structures to date. So that's the concept of the graph. It summarizes the structural states that are known either for SARS-CoV-2 or for anything detectably related to the proteins in SARS-CoV-2, summarized in one concise graph, and in terms of mimicry, hijacking, and self-assembly. And yeah, so we've also came up with a concept called a transition matrix that show this assembly of the replication and translation complex. This is the NSP7, 8, 9, 12, and 13 that I mentioned before. And the concept here is that there's quite a few different states that would occur, that could occur, combinatorially possible states of those proteins. These are the only ones that actually do occur, and the order of these states suggests quite strongly the order of assembly of the replication and translation complex. So it gives us some insights that could only really be gained by a sort of a complete study of this kind. Yeah, so that's, and I'll just skip over the details of NP12 and 14 and 16, because I think I would encourage you to look at the paper on that, and I'd like to give a bit more time to Neblina and to the demo towards the end. Okay, so with that, I'll pass over to Neblina, who will talk about the system that we've developed to enable you to use that map to navigate to 3D structures corresponding to any of the states presented.
Thanks, Sean. I'll share my screen.
Neblina.
Can you hear me? You can hear me? Okay, cool. Yep. All right, I'll start. So, hi everyone, I'm Neblina, and I'm very excited to share with you today more about the Aquaria architecture and give you a glimpse of what's happening behind the curtain. So, I'll start with some of the basic information about Aquaria. Aquaria is a web-based molecular graphics tool which was first published in 2015. It uses a structure viewer called Jollycube that renders the 3D protein structures and annotates any structural information sent from Aquaria. The main thing that sets Aquaria apart from any other molecular graphics tool is that it is primarily organized by the sequence and not the structure. What that means is that in Aquaria, a user starts by specifying a protein of interest, and Aquaria takes the UniProt sequence of the protein and finds all the PDB structure that matches to the sequence. When the user selects one of these matching structures, Aquaria aligns the sequence of the PDB structure against the UniProt sequence and annotates all the matches and differences. The part of the PDB sequence that matches to the UniProt sequence is usually shown in bright color, while the regions that don't match to the UniProt sequence are usually shown in darker colors. And when you hover over any of these residues, a tooltip displays more information about individual residue alignments. The top bar shows that the PDB structure has 99% identity to the UniProt sequence, and hovering over the question mark here will reveal more information about the sequence and structure alignment. You'll find a visual summary of all the matching structures for the sequence in the matching structures panel. Currently, there are over 1,200 structures that matches to the polyprotein 1A sequence of the SARS-CoV-2 virus. And in this panel, the structures are first ranked by the identity score and then by the length of the region that each of them covers. We also take into account the e-value, the resolution, and the experimental method that is used to determine the PDB structure to further order this sorted list. After the sorting is done, we cluster all the structures based on the region that it covers and the length of the region that it covers as well. So this is a flowchart that shows what's happening behind the scene. When a user looks up a UniProt session in Aquaria, the client sends a GET request to the node Aquaria server to find all the matching structures for the sequence. The Aquaria server then sends a query to fetch all the matching structures from Aquaria database from the PSSH2 and the PDB table. The Aquaria database returns a list of the matching structures, which is then processed in the Aquaria server. This is where the list of the PDB structures are ranked and clusters, and it is then returned to the client where it is visually rendered. We also have recently introduced client cache and server cache, which temporarily stores all the data that is required to load a matching structure for a sequence. So next time when a user looks at the same matching structure, the data is directly fetched from the cache, which significantly improves the time taken to load the structure. As you can see from this chart that I've generated, cache gives us a great performance boost. Without any cache, it takes around 10 seconds to fetch all the matching structures for polyprotein 1A. But if someone had previously looked up a protein, for a new user, that data will be fetched from the server cache, which takes less than a second. And if the user looks at the structure again, it takes less time to fetch the data from the client cache. So caching is great to ensure the best user experience. I should also talk briefly about the predefined features which are available in Aquaria. You can select any of these features and explore them on the structures. We are fetching features from Snap2, Predict Protein Cat, and UniProt, and all this data are also being cached on the client side. So now you know the complete architecture of Aquaria. So next, I'm going to talk about something very exciting. We've been working on a newer version of Aquaria called the Aquaria app, and it's currently online. So if you want to view a structure on Aquaria app, simply click on the resource button on the bottom left, and it will automatically open up the structure on Aquaria app, which will look like something like this. As you can see, we have done quite a bit of refactoring. We have maximized the space for the structure viewer. We have added the structural coverage map that shows all the dark and non-dark region for the protein of interest. You can also access all the matching structures and features data using these icons. Then we have an interactive title that will provide you more information about the UniProt sequence, the PDB structure, and the alignment information as well. And finally, we have introduced human readable URLs, which basically mean that you can look up any protein structures just by specifying the organism name and the gene name in the URL. And Aquaria will automatically find the best matching PDB structure and will update the URL automatically. Aquaria app is also designed to be supported on multiple screen sizes, including smartphones, so that your research will no longer be bound by screen sizes. And finally, I'm going to finish by letting you know that any structures in Aquaria app can be explored in virtual and augmented reality. Seán will be talking more about this, but to get started, you basically need to click on the Aquaria icon on the top left of the screen, which will reveal a QR code, and scanning this QR code on any of the supporting devices, including your smartphones, will allow you to explore the structure in extended reality. So that's all from my side. Thanks everyone for listening, and over to you, Seán.
Thanks, Neblina. All right, so I think you have to disable screen sharing. Yeah, okay. So I'll turn back my screen, and I'm going to switch now to straight to the demo. So I'm going to share Chrome. Right, so this is what the map looks like in Chrome. As I promised, all of these elements of the nodes are now links. So we can go and click on any element of the node and load the corresponding structures. Let's first start with these. So there are 14 structures where we show NSP1 hijacking the ribosomes. It's a state that I mentioned before. So I've loaded that. It does take a little while to load, even with the caching, and looks something like this. So as I mentioned before, the ribosome is quite a large structure. This is one of the really nice features of the default Aquaria coloring scheme, which is when you click on that coverage map and jump onto a structure like this, because you're coming from the virus, any part of the structure that relates to the virus will be shown with a strong coloring. So with a strongly saturated coloring, as we see here, this is the portion of this whole structure that corresponds to NSP1. The rest of it, these all correspond to host proteins, so it's the host ribosome, and they're shown initially with a semi-transparency, indicating that they are not the selected chain. So what you're currently focused on, actually, this is full thing here. This is actually polyprotein 1A, and this is the small tiny portion of polyprotein 1A where there is some structural information. That is the information that is shown in that corresponding portion. What we show down below is this is the coverage layout that Neblina introduced you to. And yeah, that's the default coloring scheme that you have that automatically draws your attention to the part of the structure that corresponds to the virus. So that's really nice in this case, where we can see the binding of this portion of NSP1 to a groove where the RNA actually threads through, and this enables it to block and control the translation machinery from that point forward. So next, I'm going to go to probably the most famous part of this graph, which is the interaction of spike glycoprotein. So we have nearly 500 structures, and of those, 46 of them in our analysis are bound to ACE2. So we can click on that, and that'll immediately load one of those 46 structures showing spike glycoprotein bound to ACE2. So these are the ACE2 3 subunits up there. And again, to explain sort of the logic, what we do in Aquaria is we, in a case of an oligomeric complex like this, we choose one chain at a time. So we see that there's actually a trimer of S protein, but we just isolate one of the elements of the trimer. And we see, of course, there are those other proteins there. So if I want to zoom back and look at the whole complex, there's a couple of ways of doing it. You can double click in the background, for example, that'll show the whole, all chains in an equal way. you could click on this. They'll actually change the view to switch the view over to looking at ACE2. So this will actually change what is focused on. So instead of looking at the spike glycoprotein, I'm now looking at the ACE2 sequence, and I'm seeing all of the structures in the database that relate to ACE2 sequence. Many of them, of course, not bound to spike glycoprotein, but this particular structure is. So, and to return, I just double click again on one of the chains associated with the spike glycoprotein to return to that view. I'd like to also draw your attention briefly to the features. So part of this work was also expanding the set of features that we enable. So we've, in Aquaria for quite a long time, we've had the UniProt features, for example, where you can look at the domains that are present in a protein. But what's, I think, particularly useful here is the ability now to extend this with some very useful features like conservation scores from the Predict Protein server. And a really interesting feature was the club, we worked together with the Cath team to not only enable the Cath domains, the superfamilies or Cath funfams to be mapped on, but actually to create these interactive pop-ups where you could actually get and interact with these pop-ups that are taken from the Cath website. So you've got really quite a lot of information available here about the functional families and even the present functional funvars, functional variants. But time doesn't permit to delve into those. I did want to just also draw your attention to one more thing, which is this link out here to our new beta version of Aquaria.app, which is the new version that Neblina mentioned. This is redesigned to make better use of screen space, particularly for sort of small devices. We still have essentially the same functionality, like that matching structures portion and so forth is available if you just by clicking on these tabs, they expand and you can access all the matching structures. But essentially, we're more or less replacing that by this concise visual summary showing the complete UniProt sequence, which part of it is covered by the current structure, and this indicates regions not in the current structure, but where there is structural information available that you can get to, and the features, of course, are also available here. And I'm going to finish with the final point about what's really exciting for us in terms of not the current paper, but the next paper that we're working on is we've been working on the ability to help with the annotation of variants. So we, as you can read here, this is what we call a human readable URL. So this is SARS-CoV-2 S protein. So as you know, it's often abbreviated just as simply S. And if you want to define a variant, you can just use a query string question mark and then a variant, or use an ampersand to put a number of variants in. And what that'll do is it'll automatically highlight that variant position in the sequence. It'll also highlight that variant position in the structure. So we can see where this particular variant lies in the structure. And what's really interesting is it tells us how many of the nearly 500 structures actually have that variant. In this case, N501Y is quite a notable variant present in some of the viral strains. And this shows us all of the structures that have that variant, either for SARS-CoV-2 or for related viral proteins. So this makes it easy to see where the variant information is available. This, by the way, also summarizes all the other variants that occur at this 501 position with bold indicating cases where there is structural information available. So there are 3 structures that have N501Q. So with that, I'm going to switch back to the presentation, which will take a little minute. And as Neblina indicated, I'm going to turn on the presentation here, and just show you what happens when you click on that or open up that QR code. So this is what you get on an iPhone or an iOS device. You get really quite a nice rendering. So basically, you don't need to install anything. This is just it's essentially your browser viewing that 3D data set. This 3D data set it can be essentially any structure. So Aquaria has, in fact, a total of 100 million models, which you can add. You can augment those models with any feature information you want, and you can view those the augmented structures in this augmented reality. This is really quite a rich augmented reality system, not just a demo system hardwired to a few simple things. How this looks, so that was the view, obviously capturing the screen. How this sort of looks when you interact with this. If you haven't tried these augmented reality demos, often you have this sort of preview mode where you can see the model. You press the AR key that opens up the camera, and then you can interact with this with the model, and it's quite nice. It's got this ability to sort of sense where desktops and surfaces are, and if you notice, there's even quite a nice shadowing effect. There's even a reflection effect as well. That's iOS. Android looks quite similar. Both devices have really sort of simple capabilities. With iOS, there's nothing to install. With Android, on modern devices, with modern Android OS, there's nothing to install. If you've got somewhat older devices or older versions of the OS, you may need to install some updates, but most devices should be able to run this mode. It's something that both Google and Apple are eager to support, so that's good for us. We actually have also support for the HoloLens. Unfortunately, HoloLens currently is limited to rather small structures just because of limitations imposed by Microsoft, total number of triangles that we can display. There's some room that we have to improve, but if you haven't tried the HoloLens, it's a much better augmented reality experience than you can get with the phone. The phone is great, everyone's got one, it's fun to play with. The HoloLens is something more like a real working device, something you can really use. So yeah, we have Android OS, HoloLens. Actually, we also can support any of the Windows mixed reality devices. This is, I think, about 20 different virtual reality devices. We even actually have a version for Sony PlayStation VR that's not easily accessible. Please contact us if you're curious about that. It's because we've got funding from the Sony Foundation to do this work. And I'm not saying that you should all start using AR and VR right now. Or XR. It's the iPhones and so forth. They're nice demonstrations. They're impressive, but they're not necessarily useful. The HoloLens, I would say, really becomes useful. It's of course more expensive. It's still somewhat an experimental device, but some of you may know that there are new augmented reality devices available on the market today. These are not available yet in every country, but they actually have, in many ways, have better performance than the HoloLens, 120 degrees field of view. And the only inconvenience, the device itself is basically the same weight as and form factor as sunglasses. The inconvenience is you've got a cable and you have to cable it to your phone currently, but that's pretty good. Now, Aquaria doesn't run on this yet. It will probably soon, but you can see that we will soon be in the position to really be using XR and AR in particular on a regular basis, and we're hoping that this technology is going to enable us to start looking at more complex structures. Sorry for the slightly silly joke, but I do think we are going to start looking at devices like this. AR has some of the benefits of looking at real physical models. The greater sense of reality can help us with understanding the more complex structures that we have to deal with compared with what they had to do with back in the 50s. Yeah, so with that, I'm just going to finish with a few quick closing remarks about how we can accelerate COVID research, because unfortunately, as you realize, many of you realize, COVID is here to stay. None of the vaccines today are going to completely eradicate this. What I'm frustrated about is the idea, and I think we've already demonstrated this in the coverage map. There's some potential therapeutic interventions that we uncovered that people have not been running clinical trials on. And I'm frustrated by the idea that we might have already gathered the data we need to make a significant breakthrough in treating COVID. Even worse, the data might have already been analyzed and it might already been presented on one of the many resources that are being hosted around the world to show these data, but the data haven't been visually organized in a way to make the insights clear. And look, it's a lot of work to build this. This took us many months to build and more months to automate and update and so forth. So I know it's not an easy process, but given the importance of COVID worldwide, I think that it's something that we need to take more seriously, which is visual organization that shows clear, reproducible patterns in our data. So many of you may know that I've been running this initiative called ISCB, which is an annual meeting series. We've had a lot of resources that we've created, and we're planning a series of discussions. We're planning a project bringing together some of the ISCB community to build insightful visualizations of COVID data. And I would encourage you to get in contact if you're interested in taking part of that. And I'd finally leave you with a closing comment, which is that, as mentioned, it's not easy to build these visualizations. One of the reasons is that we, in order to create them, we have to create something new. And there was one of our keynote speakers at ISCB who said this phrase that stuck in my mind over the last 10 years. He said, we're creating the syntax of a new visual language. We've never had data of this complexity before in general, and certainly with COVID, we've never had it. We have to invent completely new ways of organizing those data in order to make the insights clear. And there's no precedent for this. We can't really be trained in it. It's a difficult process. It requires overcoming what I would call cognitive biases, the way that we are looking and thinking about data. We have to come up with better ways. And that's a difficult step. And that's one of the reasons why we have this annual meeting series, and invite you to join us next year to be part of that. So with that, all that remains for me is to thank the organizations that have helped fund our research, the many people that have contributed, some of who are part of this call, and to invite you to join us and ask questions about our product. Thank you very much.
Thank you all very much. What a wonderful presentation. We have a couple questions. I'll do my best to read them aloud for you. Let's say I have several sequences that resembles one protein. Is it possible to create a consensus or a collective 3D model that best represents all of the sequences mentioned?
Okay, so this is not necessarily in the context of COVID. You've got, is that like, is the person online? Can they actually talk to us, or?
You can.
I think what they're asking
online, they can probably comment in the chat.
Right. I think what they're asking is perhaps the NSP3 is a case where we have this long protein. There is no structure that is available that matches the whole NSP3. What there are structures just for different domains, about 7 different domains of NSP3, where we solve structures for each of those. And there is no experimental structure for the whole. I think what the question is asking is, can we make a larger scale model that sort of brings together those different domains and comes up with a hypothesis about how they may be arranged. So there are certainly methods that do that. I think there's several recipes that I know of. Probably Chimera is probably my first port of call for something like that. I know, and Art Olson's group and David Gutzel's team have a number of recipes that you can actually run to assemble such large-scale models. It's not what we do in Aquaria. So what we do in Aquaria is we're looking at the experimental structures, and we're mapping those structures directly onto the proteome, and that's it. And then we're trying to sort of look at what that tells us.
And then the next question is it known how often the mimicry of human proteins is exploited as a means of for infection?
And again, I'm not quite sure of the context of the question. I'm assuming that the question is asked in terms of COVID-19 and SARS-CoV-2 specifically, or is it might be about viruses in general. I mean, I'll talk briefly about viruses in general, because mimicry and hijacking are well-known processes in viruses, well-known, well-described. And in terms of coronaviruses in general and SARS-CoV-2 in particular, the mimicry mechanisms that we recognized, certainly the macrodomain mimicry was well-known. Strangely, I haven't seen any evidence for the NSP13 mimicry, which I'm a bit surprised about, actually. But I haven't looked extensively, but I haven't found it in the reviews and the literature that I looked at. But yeah, the general concept of mimicry is very well known, and of course hijacking is extremely well known. Oh, I see the clarification in the comments. It was viruses in general. Yeah, I don't know. I haven't studied it extensively. So in our paper, we give a link back to a paper that discusses mimicry and hijacking, actually one for each. And you could follow up that link. It's a well-cited link, I think the best that I could find on the concept of viral mimicry. And that would probably go through a comprehensive list of which mimicries are well established. I guess the general idea is that the virus at some stage stole a gene from a host and has kept it. I think that's the general idea.